Good morning, everyone. Myself, Dr. Sakshi Bhardwaj, Application Scientist, Elton Technologies. On behalf of Elton Technologies, I would like to welcome you all for joining us today and thank you for sparing your valuable time for the session. We are excited to have you all here. This is an opportunity actually for all of us to learn from each other and grow together. As we all know, how computational approaches are booming nowadays. To get an idea of how these computational approaches are integrated with chemistry and biology, and how the software like BioWare Discovery Studio could help us in doing the invention and accelerate our research. We have our session of the day, which is entitled as Computational Approaches Are at the Interface of Chemistry and Biology. To understand this whole scenario, today we have an expert with us. Before we start the program, it is giving me immense pleasure to introduce today's speaker, Dr. Manish Kumar Gupta, sir. He is a professor from SGT College of Pharmacy, SGT University, Gurgaon. I welcome you, sir. Sir had done his PhD from CDRI Lucknow and postdoctorate research from Structure Biology Center, University of Kansas, Lawrence, KSUSA. Sir has more than 20 years of experience in the academia and research. Sir has on more than 60 peer reviewed articles, 10 book chapters, more than 2,000 citations, and one Indian patent to his credential. He has been honored with many prestigious awards too. Sir has an expert hand in molecular modeling techniques like structure based, ligand based drug designing, ligand drug interaction, receptor interaction, and molecular modeling simulations, modeling techniques, protein modeling techniques, 1D, 2D, 3D filters, and shape matchings. He is well versed in MLR, PLS, and artificial neural networks, DFT, TDDFT, and also the ab initio approaches. Sir has been working with various molecular modeling programs and got many grants in this regard. Sir is also having good knowledge of organic and medicinal chemistry. Nevertheless, Sir is also a specialist in the artificial and machine learning algorithms. Wow, this is going to be very knowledgeable and interesting session, I must say. I just request my audience to be more attentive during the session so that they can pull their queries and drop them to the chat box. So without wasting time, without any further ado, let's start our session. Up to you, sir.
Thank you, Sakshi, for a wonderful introduction.
Thank you.
Please show me my presentation. Good morning, everyone. This is Dr. Manish Gupta from SGT College of Pharmacy, SGT University. Today, I will be speaking on computational approaches at the interface of chemistry and biology. So, as Sakshi already spoke about my introduction, I did my B.Pharm and M.Pharm in pharmacochemistry and PhD from CDRI Lucknow. I did my postdoc where I learned molecular dynamics simulation of protein modeling. I have more than 20 years of experience in computational approaches on molecular design. So today I am going to discuss and show some of my experience in computational study of small molecules, biomolecules, and nanomaterials, and how modern computational technique can be used to predict their activity as well as their toxicity. So as we all know, that design approaches are broadly classified in two types of area. One is ligand-based design, where we talk about QSAR and QSTR. QSAR is quantitative exposure activity relationship, while QSTR is quantitative exposure toxicity relationship. So it can be used for both, for activity prediction as well as for toxicity prediction. Again, in ligand-based drug approach, we have pharmacophore mapping, molecular fingerprint, and here we use the approaches like artificial neural networks, PLS, MLR, and PCA. When we talk about exchange-based design approaches, we have molecule docking. That is the first approach that we can say. Then protein modeling, MD simulation, virtual screening, and database design. All these approaches are used to identify new lead molecules and for lead optimization for the development of active and selective drug molecules. So first I will touch upon a brief introduction of QSAR and QSTR modeling. It is a ligand-based approach which is used to predict the activity of the proposed set of compound. QSR models are generated by correlating the biological activity of a set of compound with their physiochemical or structural properties. And these physiochemical or structural properties are broadly classified as descriptors. This technique is used when there is no knowledge of target protein or binding site. It helps to understand the mechanism of action of compounds, and of course, it minimizes the cost and time involved in wet lab experiment for drug development process. Suppose in the previous approach, we have a series of compounds which are designed, synthesized, and evaluated for their biological activity for a defined set of target. So in this slide, you can see 25 analogs of benzyl aminobenzoic acid, and we have their biological activity at the right hand side. If someone asks you a question, what should be the 26th compound? What should be the more analogs of the same series which have more activity? So by looking at the structure itself, it is very difficult to identify what kind of molecular features would improve the activity of this particular scaffold. Here, QSR is an answer. In QSR, we convert the structure into a set of descriptors, and those descriptors are numbers. So it is rather easy to compare two numbers than compare structures to the numbers. So first we convert the structure to a set of numbers, we call descriptors, and then we correlate the activity of set of descriptors with their biological activity, which help us to develop a QSR model. Why we need QSR? The answer is, it is not always possible to practically determine all activities or property of molecules due to experimental constraint. And even if the direct measurement of activity and property are possible, they will be very tedious and time and resource intensive. Therefore, among the various approaches, the thorough studies involving the model generation is the most economical and provide fast initial assessment for further experimentation. Here, QSR depends on number one, method of descriptor generation, and also a method to establish QSR model. The methods are broadly used as MLR, PLS, PCA, and NM. Every drug molecule has some shape and surface area which helps in their cell wall penetration. And this surface area is different from different types of drug molecules. Likewise, they have flexibility which helps in its interaction with target receptor. We know when a drug combined with the target receptor or an enzyme, there is a mutual change in conformation of drug and receptor both. They also have chirality which impart selectivity of drug molecules. So one particular configuration may be biologically active while the other not. The levocitrazine would be one of the finest example where L-citrazine is biologically more active than its D form. Now come to the assumptions of QSAR. We know activity of molecules is a function of property, and property is a function of their chemical structure. It means variation in structure of molecule changes its property and hence influence its biological activity. And this is the most fundamental basis of development of QSL model. If we change the molecular structure of particular compound, we can change its property and hence biological activity. Okay, some pictures are missing on this slide. Here I have seen three types of compound: noradrenaline, adrenaline, and isoprene, and they have very slight variation on their terminal carbon. In adrenaline, we have a methyl group attached with the terminal nitrogen. In the case of noradrenaline, that methyl group is absent, while in the case of isoprene, that methyl group is replaced with a rather bulky group that is called isoprene. So isopropyl group is more bulkier than methyl group. Here we can say in the absence of methyl group, the compound is purely alpha agonist. When we attach methyl group to the noradrenaline, it becomes adrenaline. Compound shows mixed activity. It shows alpha as well as beta activity. When further we replace the methyl group, the bulkyl group like isopropyl group, compound shows selectivity towards the beta receptor. So when we increase the bulkiness from hydrogen to methyl to isopropyl, the selectivity of compound from alpha to beta increases. So here we can see if we change the functional group of the compound, we are increasing bulkiness and hence it is bring out the selectivity for the molecule from alpha to beta. It means, of course, we can bring out the selectivity in the molecule by variation in their functional groups. This is the general form of a QSR model where the activity is equal to the activity is correlated with different types of descriptors. Here X1, X2 are property of molecules, where A, B, and P are coefficient, and C is the constant. So this is an equation of a straight line, where activity is correlated with the particular property or a set of property of molecules. In QSR, we use different types of molecular descriptors. We include physicochemical, electronic, constitutional descriptors, topological descriptors, topographical descriptors. We have fingerprint based descriptors, and of course, we can use the spectral data descriptors like IR, NMR, mass, peak can be used as descriptors for developing QSR and QHDR models. Since we have a large set of descriptors, we need a very robust modeling approach to identify the most appropriate descriptors for modeling. In this slide, we can see if we have 500 descriptors and we want to develop a 2 descriptor model, we can generate more than 1 lakh QSR model. If we want to develop 3 descriptor model, we can develop more than 20 lakh models. If we want to develop 4 descriptor model, we can develop 20 billion models. Their question is, of course, we can develop billions of models, but we practically need 1 or 2 models for the design of the compound. So how to select robust model is a big question. And to answer this question, few methods have been advised by the researcher. The one method is GFA, generic function approximation, discovered by Rogers and Hopfinger. The next method was shown by Kubini. It's called museum. This is the presentation of GFA, how it works. So in the GFA, we divide the descriptor in 2 sets. We can call it first parent and second parent, and a data set of descriptor is developed by combining the property or combining the descriptor from the first parent as well as second parent. So a new set of descriptor contains the property from both parents. In the case of museum, we divide the set of descriptors into pool of descriptors where the different models are generated by using different permutation combination of descriptors. and models are selected on the basis of their R-square value and predictability. But there is one limitation of these two methods. Every time we run GFA or museum method, we got the different models because in both of these methods, the selection of descriptors in first pool is always random. Because of the randomness, we got a different set of models. This is correlated with the general population. Why the two son or daughter of a co-equal parents are different? Because the first selection of descriptor or first selection of chromosome, first selection of property is always random. Therefore, these two methods are not reproducible. To solve this problem, we came up with a method called CPMLR. This is called combinatorial protocol in multiple linear regression. This is a filter-based approach. We apply different types of filter like interparameter correlation criteria, t-value of regression coefficient, R-square value, as well as cross-validated Q-square criteria. So we apply four different filters to model, and this helps us to select the most statistically robust model that can be used for further study. The advantage of this method is highly reproducible. The advantages includes CPL generates multiple models. Models obtained in CPL procedure are unique and reproducible for a given filter conditions. The method is statistically reliable and computationally very efficient. This is example of modeling study of tetrahydrocannabinol analog for their Plasmodium falciparum protein functional transferase activity. These are the two developed models, and these are their aesthetics. We can see R value is near to 9, while the prediction of the test set is near to 7. These are the essential parameter of the new DNA compound. The figures are missing, I think. Sakshi, can you hear me? Okay, so I am continuing. The figures are missing in this slide. Figure. Fixed were there, and they were also missing in previous slide also. Okay, okay, no issue. Let me continue. I will see whether the pictures are available in the next slide or not. So, using those QSR equations, we have designed some compound and determine their biological activity on the wet lab. And here you can see the object activity is quite similar to their predicted activity by different equations. So this shows the very strong application of QSR. It helps us to design new compound. Now, QSR is the technique which were widely used in the last century. That this technique is again taken momentum due to development of different types of nanomaterial, especially metallic nanomaterial. So for the last 10 years, I have seen different types of articles, especially applying QSR based method to predict the toxicity of new developed nanomaterials. So I applied this concept to predict the toxicity of nanomaterial using artificial intelligence technique. Nanomaterial is a wide class of compound. They are different in size, different in shape, and they may have different types of toxicity. So before applying a nanomaterial for biological application, it is important to know their possible toxicity. And since we have metallic nanomaterial, they can be present in the ecosystem for the long period of time, so they may also have long-term toxicity. So before a company want to bring their nanomaterial-based formulation into the market, they have to show whether their nanomaterial are safe or not, whether they are safe for ecosystem or not. So for this, the regulatory body has recommended to you to use different types of QSR based model, and for the nanomaterial we call it quantitative nanotoxicity relationship model. So we can use these Q and TR model to build the nanotoxicity. If the nanotoxicity is below than the acceptable range, it is allowed to move into the market. If it is above the range, then again, then company has to go for the wet lab experiment to define the actual toxicity of their nanomaterials. The European Union used the REACH program, which allow the companies to use their Q and TR model for the toxicity prediction, and they are available worldwide. So they are the artificial neural network based QNAR model for toxicity of titanium oxide based nanoparticles using quantum mechanical descriptors. So we know in the artificial neural network there are several hidden layers which are used to develop the models. So here three models are shown which are developed using quantum mechanical descriptors. And in this slide, you can see the model. It is they are the highly predictive model for test set. So this shows the application of ANN based QSR modeling to model the toxicity profile of different types of nanomaterial. And this stream, as I told you, this stream is again gain the momentum. You can see different types of review articles have been published recently, which support the application of QSR based approach for the toxicity prediction of nanomaterials. And of course, you can use this technique for your research work. Now come to the second approach that is called structure-based design. This method is highly used when we have the 3D conformation of the target receptor. So SBDT is also called direct design. It is a method to design target specific drug molecules. The SBDT approach increases the probability of identification of selective ligand molecules for a particular enzyme or receptor under investigation. The target selective ligands interact specifically with their respective receptors and hence are potent in nature and show minimal side effects. So due to advancement in the biological chemistry, different types of receptors have been crystallized and their 3D conformations are available on the protein data bank. So we can utilize those crystal structures to design and develop our new drug molecule through docking study. In principle, all drug molecules act by interaction with one or more receptors and modulate the activity. Hence, SBDT methods provide an insight of various drug receptor interaction at atomic level. It relies on the 3D picture information of target receptors. This is a 3D view of a charged surface model of red PFTA receptor. It shows the different positively and negatively charged site on the receptor as well as non-polar site also. At the center you can see there is a hollow cavity where a particular inhibitor is bound. So a binding cavity is always a hollow site mainly containing hydrophobic amino acid residue where our hydrophobic molecules bind. Where the charged residue of a particular receptor are exposed to the solvent. The binding cavity is mainly contained hydrophobic residue and it is always in a groove form of shape. Let's talk about the docking. Docking is a method to identify the optimal conformation of the ligand at the receptor site. In docking, multiple dot conformation of a ligand are developed, and their binding efficiency in terms of scoring function is calculated. The conformation associated with a reasonable scoring value is considered as putative biaxial conformation. So in the docking, we don't get one conformation. We explore different conformation of a ligand, and then we identify their binding efficiency in terms of scoring function. We select the most putative biaxial conformation on the basis of favorable drug receptor interactions. These are the some potent ligands which have been identified using SBDD approach, and you can see all these potent compounds are showing activity at nanomolar range. If the activity is in the nanomolar range, we can say okay, the drug may have activity, a drug may have dose in some milligrams, 5 milligrams or 10 milligrams. How SBDD is helpful in designing of target specific drug molecules. Now see this example. So I'm sorry, some figures are not appearing in this presentation. Here I have given the example of acetylcholine as well as adrenaline. Acetylcholine is a linear molecule, while the adenine and noradrenaline they contain a cyclic aromatic ring structure. The moiety is called catechol. Acetylcholine never interacts with the adrenergic receptors. Adrenaline and noradrenaline, they never interact with the acetylcholine receptor. They are the biological hormone present in our body. Why they do not interact? Because they have the very specific features which are complementary to the receptors. So through the docking study, we can impart the selectivity to the molecule by considering the binding cavity, binding cavity, and amino acid residues present in the binding cavity. So we can see the amino acid residue, we can see the binding site, we can see the nature of the different types of residue, and of course we can design our molecules by bring out the complementarity in the functional group. How I will show in the next slide. This is the dot conformation of a ligand at the xanthine oxidase. This is probably my favorite slide because here I can show you almost all types of drug receptor interactions. At the extreme left, you can see methyl moiety of the drug is interacting with the two aromatic residue of the receptor. The one is phenyl 914, the second is phenyl 1009. They are interacting with the drug molecule via pi-pi stacking interaction. And interestingly, you can observe two types of pi-pi stacking interaction. The one is face-to-face pi-pi stacking. The next one is face-to-face pi-pi stacking. 914 PHE 914 is involved in the face-to-face pi-pi stacking interaction with the inhibitor, while the PHE 1009 it shows edge-to-face pi-pi stacking interaction. So these two interactions are very, very important to stabilize an inhibitor at the receptor binding site. The next interaction is H1 interaction shown by the glutamine 802. Now we can see a hydrophobic interaction of the terminal alkyl group of leucine 1014 with the CH2 group of the inhibitor. We have again a alkyl type van der Waals interaction with the threonine THR803 with the terminal methyl group of the inhibitor. We have seen a very interesting cation pi interaction with the lysine 771. So lysine is positively charged at the receptor site. It is interacting with the circulating pi electrons of the aromatic functional moiety. So in this slide, we can see majority of the important drug receptor interaction which are helpful to stabilize an inhibitor at the target site. Now come to the MD simulation. So most people say, okay, since we have dot conformation of an inhibitor at the target site, we can design a new compound, but there is a limitation. Limitation is when a drug binds with the receptor, there is a mutual change in conformation. It means we also observe the change in conformation of the receptor. While in the docking study, there is a provision to change the conformation of the inhibitor, but the receptor is always static. Do not allow the target receptor to move in the docking. To solve this, once again, a drug molecule binds the target receptors at the particular pH, particular temperature, and at the particular pH and temperature, the conformation of the receptor is different from the pH and temperature at which the receptor is crystallized. So when the receptor is crystallized in the wetlet, there is always change in conformation of the receptor due to change in pH and change in temperature of the medium. Therefore, MD simulation technique is used which help us to determine the change in conformation of the receptor when it interacts with either its substrate or inhibitor. So, MD simulation is an in silico method of of the physical movement of atoms and molecules in presence of solvent or in vacuum. Following the laws of classical mechanics, it provides a detailed description of atomic motion. In this, the atoms and molecules are allowed to interact over a period of time at given temperature and pressure. MD simulation provides detailed information on the fluctuation and conformational changes of protein arising due to its interaction with substrate ligand, while this information is not available in documentation. As I told you, the drug receptor interaction is a dynamic process which is often associated with change in conformation of drug and receptor both. The docking software usually provides the procedure for the flexible docking of a ligand at the static binding site of a receptor and hence does not allow the study of change in conformation of receptor during drug receptor interactions. Hence, MD simulation study is very helpful to study the effect of drug receptor interactions on the change in conformation of receptor or binding site residue. Also, the 3D conformation of a receptor is determined at the pH, solvent, and temperature, which is different from the body. Therefore, the X-ray conformation of a receptor is not an assurance of its conformation in a biological system. You will see always a difference in the X-ray conformation and biological conformation of a receptor. Here, MD simulation study helps to understand the conformation of a receptor at physiological pH, temperature, and pressure. Here I will give you some examples where I have applied the MD simulation study. This is an example of MD simulation study of neurokinetic receptors. It is a G protein receptors. When I did the MD simulation study of receptor, the PD conformation was not available, so I modeled this receptor through homology modeling. So, if the target receptor is not available from the protein data bank, homology modeling is a technique to determine the possible 3D conformation of the target receptor. So here I applied the concept of homology modeling. So here we study the similar receptors for which 3D conformation is available, and by utilizing the 3D conformation of the available receptor, we determine the conformation of unknown 3D conformation of unknown receptor. So for the neurokinin one, I have taken different templates like beta 2 receptor, adenosine receptor, bovine rhodopsin receptor, and dopamine receptor to develop our neurokinin receptor model. These are the 3D conformation of NK1 receptor model. This is the MD simulation system. Since neurokinin receptor is a G protein coupled receptor, it is a transmembrane receptor. So we have developed a membrane environment surrounding these receptors. So it contains phospholipids, water molecules, ionic bodies similar to those found in our cell walls. So at the left hand side it is the frontal view, right hand side it is the top view. The study was carried out few years back. At that time, the computational capacity our system was not very good, so we run this particular simulation for 4 nanoseconds. You can see the green is the simulation, green line is simulation of ligand, red is protein, and blue is the protein ligand complex. This picture shows that when a drug bound with the protein, the flexibility of protein is reduced because the residue which were more flexible when the drug was not bound, show high degree of deviation, high degree of fluctuation. When drug is bound, the flexibility of the residue is reduced. So it shows how tightly a drug is bind. When there is more binding of the drug with the target receptor or with the binding residue, the flexibility decreases. This picture shows the root mean residue fluctuation of residue. So on the right hand side, the graph shows when there is no drug bound with the neurokinin receptor. On the left hand side, it is the MD simulation of protein ligand complex. In the terms of RLSG value, the fluctuation is more when the receptor is in the free form. When the drug is bound with the receptor, the fluctuation is less. Again, this is a diagram of 2D depiction of drug receptor interaction. On the left hand side, you can see the drug receptor interaction before MD simulation. We can hardly see one or two interaction with of the drug and receptor. On the right hand side, with the figure after MD simulation, in clearly it clearly shows drug receptor interaction are clearly visible. They are more apparent when we study their interaction after MD simulation. So in this case, during the MD simulation, the amino acid residue which were important for their interactions are moved toward the drug molecules. They have shown the flexibility, they have shown the inclination for the drug molecule, and we have identified some useful drug receptor interactions while those were absent before MD simulation. They are the movement of different residues before and after MD simulation. They show the flexibility of different types of residues. This is the MD simulation study of DNA topoisomerase 1 DNA complex. The figure A is binary complex, the figure B is the ternary complex. Ternary complex means topoisomerase, DNA, and drug molecules. So when a drug molecule binds with the receptor, its flexibility decreases. The same is appeared in the figure number B, where the receptor is less flexible in comparison with the binary complex. So if a drug binds tightly with the receptor, its flexibility decreases. So when you have two or three drug molecules, and of course we have two ways. First, we can calculate the binding energy of the drug receptor interaction, and second, we can also consider the flexibility of the receptor. If the drug is tightly bound, we will observe the less flexibility in the receptor. This is the MD simulation study of HIV integrase in the presence and absence of drug molecules. On the figure left hand side, you can see this residue which is shown in yellow and red is flexible, while in the case when the drug is bound with the receptor, this flexible residue tightly covered the drug molecule and involved in important drug receptor interaction. The drug molecule is tightly packed between helices and flexible loop. It is clearly visible in this slide. Now I extended this activity not only for the biomolecules from the human, I also applied my MD simulation study for plant hormone and plant receptor. So ABA is very important plant receptor which interact with the abscisic acid. Abscisic acid is a plant hormone which play very important role during the drought. When in the case of drought, this particular hormone is activated and modulate the stomatal closure in the plant leaves. So mainly regulate the plant water use in the plant. So I was working on the project. The object was to develop a new type of AVA agonist that can help farmers to protect their plant in the drought condition. So what we observed is this particular agonist bind in a very unique fashion with a target receptor. On the right hand side, you can see on the figure B, there is a L4 loop which is called gate, L5 loop which is called lash, and there is a alpha 4 loop. So these L4, L5, L2, and alpha 4 they make a binding cavity for ABA receptors. So when ABA is not present in the plant system, this gate loop is open. When ABA interact with the receptor, this gate loop is closed and less is function in its position. I will show the next slide. This is a parabactin. Parabactin acts as an agonist for, there are three types of ABA receptor, even there are 12 types of ABA receptor in the plant. I confined my study for three types of ABA receptors, ABA receptor 1, 2, and 3, and these receptors were identified from the rice. This parabactin act as an agonist for ABA receptor 1 and antagonist as ABA receptor 2 and partial agonist at ABA receptor 3. So objective was why one compound parabactin acting as a agonist for receptor 1, antagonist for receptor 2 and partial agonist for receptor 3. So we have carried out the anti-simulation study of ABA 1, 2 and 3 receptors in presence of parabactin, and we can see the different conformation of L4 gate loop in these receptors. You can see in the figure A, the loop is tightly closed. In the figure B, the L4 loop is open. It means the parabactin can occupy the binding site, but it failed to induce the favorable change in conformation of the receptor, which it further failed to induce the downstream signaling. That's why it is acting as an antagonist. Again, in the figure C, it is tightly bounded, but the L2 loop is not supporting, is not in the same conformation as in the case of figure number A. So in this case, MD Samuel study help us why one compound is acting as an agonist for one receptor and antagonist for another receptors. They are the RMSD value of all three receptors in the presence of ABA and parabactin. You can see bottom two figures, figure number A and figure B. Figure A, we have taken the ABA, that is a natural substrate, and parabactin, that is shown in the red. This figure shows that APA and pyrobactin, they show the similar change in conformation of the receptor, and that's the pyrobactin acting as agonist. It mimics the activity of ABA. While in the case of graph number B, different residues, it shows the flexibility of different residue. It is not same as the plot number A. Its ABA, pyrobactin behave or interact differently with the ABA receptor 2 as well as ABA receptor 3. It is showing agonist activity in ABA receptor 1, while the antagonist activity or partial agonist activity for ABA receptor 2 as well as 3. So here we can see how MD formulation help us to understand the mechanism of action of one drug molecule on different types of receptors. Now come to the conclusion. We have seen QSAR approaches are used efficiently in the prediction of activity of new compounds prior to their synthesis. You should predict environmental toxicity of compound as well as nanomaterials, genotoxicity, ecotoxicity. It is also used to predict the reactivity of organic and inorganic pollutants, reactivity of oxidant and free radicals. QSR approaches are used in in silico property prediction of new compounds, which are very helpful to select appropriate material for the synthesis of or preparation of new chemical system. Now come to the SBDT approach. It is a rational and affordable approach to design target specific drug molecules or inhibitors. The docking models are used in virtual screening to identify new leads to target the receptor under investigation. They are used to development of more efficient scoring functions are required to minimize false positive. That's why different docking software use different types of scoring function, and development of more and efficient scoring function is in progress. There are some docking softwares are also available which provide some degree of flexibility in the receptor and amino acid residue of the receptor. MD simulation tree, which considers the flexibility of receptors, are very useful to understand the mechanism of enzyme activation or inactivation by inhibitors. Thank you so much. This is my favorite slide where you can see the thank you in different conformation. So visually we can identify the thank you at the center is the correct one. Thank you so much. Ms. Sakshi, the session, can you take
Thank you
the
so much, sir.
question?
Here, that thank you
Now,
slide is like, giving the, exact picture how the ligand and receptor interaction will show the different conformations. So that, thank you so much, sir, for such a wonderful and the knowledgeable presentation. I must say that our audience will definitely learn more about these computation, how these computational approaches are integrated or how they are at the interface of chemistry and biology, how we can come back with the chem informatics and the bioinformatics with the computational approaches, how we can take the advantage of like softwares like bioware discovery studio to integrate or to help us to design the new entities which could be a drug for different kinds of therapies, how we can study the pharmacophoric features, how we can study the generate the QSAR models of those ligands, and those models we can utilize for designing of different target molecules or the drug ligand molecules. You clearly said how the docking is a one part, and then getting into the simulation is a different part where we can study the stability and flexibility of our ligand receptor interaction, how much in a real time environment how they are behaving, how fluctuating, how much there is a fluctuation in between the ligand and protein. So it is well said, this thing to the audience. Thank you so much. We have some questions with us. So the first question is, what is the difference between the structure-based drug designing and ligand-based drug designing?
right. So I'm happy to answer this question. So when we have the 3D conformation of the receptor. It is always good to adopt the structure-based design because we have the 3D conformation of the target receptor. Now, like on the counter side, if we do not have the 3D conformation of the target receptors, then we can go for the ligand-based design, which include QCR and pharmacophore model. So when we have the target receptor, go for the HSF based. When we do not have the target receptor, go for the ligand based. We consider the property of the already reported molecule to design the new set of things. This is called ligand based.
Well said, sir. Next question. Why machine learning is important in drug discovery?
Again, very interesting question.
Yes.
When we use the computer, when we sit on the computer, of course, we can deal with the some compound which can even draw on the computer system and can be optimized. But last a few years, we have some equals there. For the last few years, we have observed that different database of compounds have been developed by the different scientists. The zinc database of compound is one of the examples. So we have millions of compounds are available. And if you want to identify some compounds from that database for your target receptor, you cannot do manually because there are billions of compounds. When you want to dog this compound on your receptor, you have to minimize this compound for their energy. You have to correct their geometry. You have to correct their charges. You cannot do it manually. In this case, we use the advantage of artificial intelligence, which help us to optimize the molecular system as per the command given. All the molecules are optimized and they are dogged by using the artificial intelligence. So the advantage is using artificial intelligence, you can deal with the billions of compounds at the same time. Your mic is Sakshi.
Yes, sir. Can we use same kind of QSR for toxicity predictions?
Yeah, of course. For the QSR, we compare the property of the compound with the its activity. The activity may be toxicity also. Activity means suppose you can say ki, okay, anti-cancer activity, cholinergic activity. So toxicity means if the compound have IC50 value. IC50 means the compound is able to kill the. It is a concentration which inhibits the activity at the 50% of the cell population. So it can be also for toxicity as well as activity. Both can be done. Because both are the number. Both can be given in terms of IC50, MIC and all. So it can be done.
Okay, thank you, sir. Uh, Next question is how to conclude which compound is stable with drug target during the dynamics?
Yeah, very interesting question. So the question
Most
is,
popular question, I
I
must
think,
say.
what do you want
Yes.
to, what do you want to know? Stability of the compound or stability of the receptor complex? So to answer of this question, we have two types of approaches. First, we calculate the RMSD, root mean square deviation, which come out from the MD simulation. So here MD simulation, we study the movement of receptor in the presence of drug. I am repeating. We study the movement of receptor in presence of drug. So if the drug is tightly bounding, you will see the less fluctuation in the binding site residue. And that is visibly clear when you calculate the RMSD value and plot the graph with respect to the time scale. Second is you can also plot the root RMS. Root means square fluctuation of each individual residue. Which particular residue is fluctuating with respect to the time. So here you can identify which residue more showing more fluctuation, and the same residue, if bound with the particular inhibitor, what is its degree of fluctuation. So what we observe it when the in the case of drug receptor interaction, the fluctuation of residue is less in comparison with the receptor when it is free form. So can be used calculating can be determined using calculating RMSD value as well as RMS value.
Thank you. I think are we having more questions? Okay, thank you so much, sir. It was a very valuable session, and thank you for the great presentation. It was a pleasure to have you with us, sir. Great, thank you all. We appreciate you for being with us today. Thank you so much. Thank you, sir. Thank
Thank
you.
you, Sakshi, and thank you, Elton, for giving me the opportunity for this presentation. Thank you so much.
Thank you. Thank you so much. Thank you.